Welcome back, everyone. So we're going to continue on with Chapter 8, Molecular Biology. So in this third video, we're going to get into transcription and then the fourth video, translation. So this basically takes our genetic information, specifically our genes, and we're going to take that information and we're actually going to make things with it. So we're going to take our genes and learn how we can use it to code for our different traits through protein synthesis. Okay, so we already learned about, again, that in the last video, how one of the functions of DNA is replication. So the second function of DNA is to basically, again, provide the information needed to make proteins. And so the first step of that is transcription. Okay, so we're going to learn about the first step of that protein synthesis being transcription, which is basically where DNA is read or transcribed into an mRNA molecule. And then the mRNA, so that's basically the code that helps form a protein through a process known as translation. So in this video, we're going to walk us through the process of transcription. So we'll learn by the end of the video, you'll be able to explain what the central dogma is and explain the main steps of transcription. And then there is a difference between how eukaryotic mRNA is processed and then basically the termination of transcription in prokaryotes. So you'll be able to describe that. Okay. So let's start out by just taking a look at a summary. So transcription, again, with DNA, DNA, as we stated, DNA provides the information needed to construct the proteins necessary for the cell to perform all the functions. So in the next two videos, we're going to learn this process. So if you think about it, here's a very simplified view of a cell. So we have our nucleus here, and remember how the DNA is housed inside the nucleus. And the DNA contains, thousands of genes on it, and each gene will code for a certain protein, okay? But the DNA stays in the nucleus. Okay, so the DNA is protected by the nuclear envelope. It stays in the nucleus. But the ribosomes, which are the protein factories, are out in the cytoplasm. So how do we get the information to the ribosomes? Well, we have something called mRNA. So mRNA, think of mRNA, that M stands for messenger. So the mRNA is the messenger. So it takes the information that's coded on the DNA and it is transcribed into the mRNA, the message. It carries that message out into the cytoplasm. So it carries the code. So it takes that code and it carries it out into the cytoplasm where it can form the protein through it can provide the code to form the protein through translation. Okay, so DNA never leaves the nucleus, the ribosomes never enter the nucleus, so the messenger is the mRNA. Okay, so it takes the message from the nucleus out into the cytoplasm and tells the ribosome what to do, what to make. Okay, and here's the amino acid sequence that's going to make that will eventually become the protein. All right, so let's get into the whole process. So first off, let's talk about the central dogma. So the central dogma states that DNA encodes RNA and RNA encodes protein. Okay, so that's it. Okay, the central dogma of biology is this huge, big deal. The central dogma of biology. It's very simple. DNA encodes RNA, RNA encodes proteins. Okay, which basically means that the genes, our individual genes, they specify the sequences of the mRNAs, which in turn specify the sequences of the proteins. So if you think about it, like I said, the DNA is in the nucleus, the proteins are being made out in the cytoplasm. So we need the RNA, put a little M inside, specifically the mRNA, to be the middleman. Okay, so the middleman is taking the instructions from the DNA and telling the ribosomes how to make the protein. Kind of like, I like to think of it as like the DNA is kind of like the cookbook, and the RNA is a recipe from the cookbook, and the protein is the chocolate chip cookie that you end up making. Okay, so here's the cookbook has all the recipes in it. You only need one recipe to make the chocolate chip cookies. You just need the chocolate chip cookie recipe. So the RNA is the chocolate chip cookie recipe that you copy down from the big cookbook, and then you take that chocolate chip cookie recipe and you make the cookies from it. Okay, so here's the physical cookies that are made. Okay, so that's how I think of it, as the DNA is the cookbook, the RNA is the specific chocolate chip cookie recipe we're looking for to make the chocolate chip cookies. All right, so that's how I think of it. So that's the central dogma. So the DNA encodes the RNA encodes the protein. Okay, so we're going to start off with, it's actually a crash course video it basically goes into both transcription and translation. So even though our lecture video here only covers 8.3 transcription, realize we're going to get into translation in our 8.4. So it covers, so this will be an overview of both. But Hank goes into both, the central dogma that we just talked about, as well as transcription and translation. So it's a great overview, and then we'll get into the details.
Okay, roll it. You know what this is? It is the longest word in the world. Like anywhere, any language, more than 189,000 letters. If you were to write it down, though I don't know why you would, it would fill up more than 100 pages, and if you could actually say it without, breaking your face, it'd take about 5 hours. What the frick is this word? Well, it is the name of the longest known protein on Earth, and it's actually in you right now. Because of its enormous size, it was given the name Titan by scientists, and that's with 2 Y's. And it's a protein that helps give some the springiness to your muscles. Today we're going to be talking about DNA and how it, along with 3 versions of its cousin RNA, unleash chemical kung fu to synthesize proteins just like this. So this is going to take a while to explain. So how about we make ourselves some Hot Pockets? Oh, my favorite, ham and cheese. Every time I take a bite, I wonder, how did they do it? How did they pack exactly the same flavor into every foil cardboard wrapped food-ish item? Clearly there's got to be some super secret instruction manual kept and a location known only to 2 people. And since I'm talking about biology here, that brings up a related question. How did I get built from the DNA instructions and biological molecules we've been talking about? Today, that's what I'm going to do. Not actually make Hot Pockets or a person, but I'm going to be talking about DNA transcription and translation, which is how we get made into the delicious things that we are today, though hopefully none of us know how delicious people are. Animals, plants, and also Hot Pockets really are nothing more than salty water, carbohydrates, fats, and proteins combined in precise proportions following very explicit instructions. Let's say I want to make my own Hot Pocket. I would have to, 1, break into the lair of the Hot Pocket company holding the secret manual. 2, read the instructions on how to make the machinery to produce the Hot Pocket and the proportions of the ingredients. 3, quickly write down that information in shorthand before I get caught by the Hot Pocket police. 4, go home, follow the instructions, build the machinery, and mix the ingredients together until I have a perfect Hot Pocket. And that is how we get us. Very simply, inside the cell's nucleus, The DNA instruction manual was copied gene by transcription onto a kind of RNA and then taken out of the layer where the instructions are followed by the process of translation to assemble amino acid strings into polypeptides or proteins that make up all kinds of stuff, from this titan down here to the keratin in my hair. But most of the polypeptides that get made aren't structural proteins like hair, they're enzymes which go on to act like the assembly machinery, breaking down and building and combining carbohydrates and lipids and proteins that make up variations of cell material. So enzymes are just like whatever ingenious machinery they use the factory to make this. Okay, let's start out in the layer. The nucleus. The length of DNA that we're going to be transcribing onto an RNA molecule is called our transcription unit. And let's say in today's example that it's going to include the gene that transcribes for our friend Titan, which in humans at least occurs on chromosome 2. Now each transcription unit has a sequence just above it on the strand, and that's called upstream. Biologists call that upstream on the strand, and that sequence sort of defines when the transcription unit is going to begin. This special sequence is the promoter, and it almost always contains a sequence of 2 of the 4 nitrogenous bases that we talked about in our last episode: adenine, thymine, cytosine, and guanine. Specifically, the promoter is a really simple repetition. We got thymine, adenine, thymine, adenine, and then A, and then on the other side, AT. Because you know how this works, right? This is called the ta-ta box. It's nearly universal, and it helps our enzyme figure out where to bind to the strand. Now you'll remember from our episode about DNA structure that DNA strands run in one of 2 directions, depending on which end of the strand is free and which end has a phosphate bond. One direction is 5' to 3', and the other direction is 3' to 5'. In this case, upstream means toward the 3' end, and downstream means toward the 5' end. So the first enzyme in this process is RNA polymerase, and it copies the DNA sequence downstream of the ta-ta box, that's towards the 5' end, and copies it into a similar type of language, messenger RNA. Quick aside, so you'll notice that to read the DNA in order to make enzymes, we need an enzyme in the first place, so it kind of gets chicken egg here. We need the enzyme to make the DNA and the DNA to make the enzyme. So where did RNA polymerase come from in the first place if we haven't made it yet? What an excellent question. It turns out that all of these basic necessities get handed down from your mom. She packed quite a lot more into her egg than just her DNA, so, we had a healthy start. So, thanks Mom. So the RNA polymerase binds to the DNA at that ta-ta box and begins to unzip the double helix. Working along the DNA chain, the enzyme reads the nitrogenous bases, those are the letters, and helps the RNA version of those nitrogenous bases floating around in the nucleus to find their match. Now you might also recall from our previous episodes that nitrogenous bases only have one counterpart that they can bond with, but RNA, which is the pink one here, doesn't have thymine like DNA does, which is the green and the blue. Instead it has uracil. So U appears here in T's place as a partner to adenine. As it moves, the RNA polymerase rezips the DNA behind it and lets our new strand of messenger RNA peel away. Eventually the RNA polymerase reaches another sequence downstream called a termination signal that triggers it to pull off. Now, some finishing touches before this info can safely leave the layer. First, a special type of guanine is added to the 5' end. That's the first part of the mRNA that we copied, and that's called the 5' cap. On the other end, it looks like I fell asleep with my finger on the A key of my keyboard, but another enzyme added about 250 adenines onto the 3' end. This is called our poly-A tail. These caps on either end of the mRNA package make it easier for the mRNA to leave the nucleus. They also help protect it from degradations from nearby passing enzymes, while also making it easier to connect with other organelles later on. But that's still not the end of it. As if to try to confuse me to protect the secret Hot Pocket recipe, the original recipe book also contains lots of extra misleading information. So just before leaving the nucleus, that extra information gets cut out of the RNA in a process called RNA splicing. And it's something like editing a video. This process is really complicated, but I just have to tell you about 2 of the key players because they have such cool names. One, the SNRPs, which are small nuclear ribonucleoproteins. These are a combination of RNA and proteins, and they recognize the sequences that signal the start and end of the areas to be spliced. SNRPs bunch together with a bunch of other proteins to form the spliceosome, which is what does the actual editing, as it were, breaking the junk segments down so their nitrogenous bases can be reused in DNA or RNA, and sticking together the 2 ends of the good stuff. That good stuff that gets spliced together, by the way, are called the exons because they will eventually be expressed. The junk that gets cut out are just the intervening segments, or the introns. The material in the introns will stay in the nucleus and get recycled, so for instance, Titan down there is thought to have hundreds of exons when it's all said and done, probably more than 360, which may be more than any other protein. And it also contains the longest intron in humans, some 17,000 base pairs long. Man, Titan, it is just a world record holder. So now that it's been protected and refined, the messenger RNA can now move out of the nucleus. Okay, so a quick review of our Hot Pocket Mission Impossible caper so far. We broke into the layer containing the instructions, we copied down those instructions in shorthand, we added some protective coatings, and then we cut out some extra notes that we didn't need, and then we escaped back out of the layer. Now I have to actually read the notes, make the machinery, and assemble the ingredients. This process is called translation.
Okay, so that was a great introduction into transcription. So we're going to get into all of the specifics of transcription. So in your textbook, they divide transcription into 3 main stages. So initiation, elongation, and then finally termination. So we're going to do the same thing. We're going to start with the initiation stage, and then we're going to move on to elongation and then termination. So let's start with initiation. Okay, so in initiation, we saw from the video how there's a certain sequence called the promoter that is where everything starts. Okay, so this is where we have basically a transcription bubble starts to form, and that transcription bubble will be where the double helix partially unwinds, and something called RNA polymerase, which is an enzyme, will bind to the promoter sequence on the DNA strand. Okay, so the promoter sequence, like what we saw from the video, is a sequence that is very specific. Okay, so it starts with TATA. Okay, so it's called the Tata box, right? So TATA. So it's easy to remember. And so, you know, it says TATA, and then it was TTTT, okay? But you don't need to remember that. So it's just remember the Tata box. So that's a specific sequence. So that's where the RNA polymerase knows where to bind to, okay? And it's upstream from the actual gene that needs to be transcribed. So the RNA polymerase is going to bind to that specific area or sequence that, and it knows, okay, that's where we're going to start the process. Okay, and that's it for initiation. And you'll notice, you know, initiation, elongation, and termination. Hey, that's the same as the 3 names of the 3 stages and DNA replication. So you know, it'll be kind of easy to remember. So that's it for initiation. So basically we have the RNA polymerase binding to the Tata box, which is the promoter sequence on the DNA strand. And that forms the transcription bubble. Okay? So next, we get elongation. So same process, you know, same sequence as in DNA replication. So in elongation, notice we have 2 strands of DNA. We have the DNA, what's called the DNA template, and we have the non-template strand. So the RNA polymerase is going to be reading the DNA template strand. Okay, so it's only going to be following one of the strands. It's not going to be following both. So it's going to be reading the DNA template strand, and it's going to be reading it basically from the 3', reading it from the 3' end to the 5' end of the template strand. And remember, just like in DNA replication, the RNA polymerase, just like the DNA polymerase, is going to be adding RNA onto the 3' end. Okay, so remember, just like DNA polymerase only adds to the 3' end. Okay, so it's building off of the 3' end. Okay, so it can only add on to the 3' end. Okay, so mRNA is synthesized from the 3' end. Okay, and if you notice, if we look at the as it's being made here, this mRNA strand, it's forming complementary base pairs to the template strand. So if we look at thymine on the DNA strand, okay, an adenine will be placed on the mRNA strand. If we have a guanine on the template strand, a cytosine will be placed on the template strand. If we have a cytosine on the template strand, a guanine will be placed on the RNA strand. Okay, so it'll be complementary base pairing. Okay, the only difference is, remember, RNA does not have thymine. So if there's an A on the DNA strand, then a uracil will be placed on the mRNA strand, okay, because there's no thymine on RNA, so it has to be uracil instead. All right, so as this is being produced, okay, once it's being produced, as the RNA polymerase is moving through, okay, the DNA behind it will get rewound, and then the DNA ahead of the transcription bubble will be unwound. Okay, so this is just moving through this little transcription bubble will be, unwinding, and then it's immediately rewound past the bubble and unwound in front of the bubble. Okay, so it's pretty simple, and you can see that this mRNA strand, it's pretty much identical to the non-template strand. Okay, so if I look at this this strand here, it looks, so we have C, C, A, C, C, G, G, C, C. It's pretty much identical to the non-template strand, except for instead of thymine, there's uracil in it. Okay, so the mRNA strand is identical to the non-template strand with the exception of uracil in place of thymine. All right, so it's being built off of the template strand, but it looks just like the non-template strand with the exception of the uracil there. All right, so elongation will just keep on going all the way through through the sequence of the gene. Okay, so I'm going to talk about termination for the prokaryotes first, and then we'll talk about the termination in the eukaryotes. Okay, so with the prokaryotes, we have basically what ends up happening is once a gene has been fully transcribed, we end up having in prokaryotes, there's a termination signal. So just like in the video, we saw, at the end of the gene, there's some sort of termination signal, which is just like an enzyme involved, which is we have this repeated sequence of DNA, and that signals the end of the process. So we're done with the RNA. And so what ends up happening is that the RNA polymerase will release from the DNA template, and so the DNA template and the mRNA are separated from each other. Okay, and so we're done. Termination, the transcription has been completed. But what's interesting that's different between prokaryotes and eukaryotes is that with eukaryotes, remember, transcription is occurring inside the nucleus of the cell, and the ribosomes are in the cytoplasm. So transcription and translation, they occur separately from each other. But with prokaryotes, there is no nucleus. So transcription and translation can occur simultaneously. So while the transcript is, while transcription is occurring, the part of the transcript that's already been transcribed the ribosome can already start attaching to it and translating it. Okay, so we can already start making protein while transcription is still occurring. So that makes protein synthesis occur really, really quickly in prokaryotes, okay? Whereas in eukaryotes, it takes a lot longer because transcription has to occur and finish inside the nucleus, and the mRNA has to leave the nucleus and then attach to a ribosome, and translation has to occur completely separated, okay? So a lot different process taking place, okay? So with prokaryotes, you see these ribosomes, they're attached already to the mRNA while the mRNA is being transcribed, okay? So the mRNA is being transcribed, the translation is already occurring while it's still being transcribed. Okay, so those proteins are already, partially being made while, it's still in the transcription process. So that's pretty neat. Okay, so that just means that the proteins, they can be made pretty quickly inside a prokaryotic cell versus a eukaryotic cell. All right. All right, so let's move on. Let's go back to the eukaryotes. With the eukaryotes, just like with the prokaryotes, there is a termination signal at the end of the gene, which is just a sequence of DNA, just like what we saw with the prokaryotes. But with the eukaryotes inside the nucleus, we're not done yet, okay? The mRNA still needs to go through what's called RNA processing before it can leave the nucleus and attach to a ribosome for translation to take place, okay? So first off, the mRNA transcript, okay, this mRNA strand needs to be coated with some RNA stabilizing proteins, and then that will help prevent that mRNA strand, once it leaves the nucleus, from being attacked by enzymes and degraded. Okay, so one thing that gets placed is it receives what's called the 5' cap. So at the end of the mRNA strand, it receives this 5' cap. So that helps, again, it helps prevent it from being degraded by some enzymes, but or whatever, out in the, lysosomes and things like that, out in the cytoplasm. But it also helps when the ribosome comes along, it knows where to attach, okay, which end to attach to. So it'll recognize, okay, here's where I'm supposed to attach to this side. I'm not going to attach to that end. Okay, so the ribosome knows where it's supposed to attach to, okay, and that's where it knows where to initiate translation, okay? So that receives the 5' cap in order to allow for, the initiation of translation. The other end, the 3' end, receives what's called a poly-A tail. And we saw from the video, the poly-A tail is just basically a bunch of adenines attached. And that again is just, it could be like 200, I think the video stated 250 adenines long. Okay, so it's just a bunch of A's. And that again is just to prevent further degradation. So it's just to, tell the lysosomes and the other, enzymes that are floating around in the cytoplasm that, this mRNA is supposed to be there, don't break it down, it's supposed to be there, it's just heading, to find a ribosome, so don't break it down, okay? So that's the first step of RNA processing. So the mRNA strand, so this is the mRNA strand, okay, it needs to get a 5' cap so that the ribosome knows where to attach to start translation, and a poly-A tail, okay? Next, that RNA strand, that mRNA strand, has two parts to it. It has what are called exons and what are called introns. So exons are protein coding mRNA sequences. Okay, so they actually code for protein. Introns are non-coding, non-protein coding, what we call intervening sequences. So we call that kind of junk DNA, or junk DNA sequences. So we need to splice those out before the mRNA leaves the nucleus. Okay, so from the video you saw how there's an enzyme called a spliceosome. So the spliceosome is going to splice the introns out. So the introns need to be removed. So think introns are the intervening parts of the mRNA. So those have to be spliced out. So they get removed. Whereas the exons are the parts that get expressed. So they need to stay. So the introns get spliced out, the exons get glued together, and theirs are spliced together and will be expressed. All right, so those are all nice and ready to go. So once the introns are removed, they get degraded, and then the mRNA is fully processed, ready to go. It's got its poly-A tail to prevent the mRNA from being degraded. It's got the 5' cap so that the ribosome knows where to attach to. Okay, so now it's fully processed, it's ready to go. All right. So you might be wondering, why do we have introns in the first place? For a long time, scientists didn't really know. They just called it junk DNA. But recent studies, they actually think there's a purpose for it. They think it has to do with, maybe it has to do with gene regulation. So there is a purpose, and they're still learning about it. Okay, so we do think there is a purpose for those introns. Okay, all right, so let's move on to some review questions. So a promoter is A, specific sequence of DNA nucleotides, or is it B, a specific sequence of RNA nucleotides, or is it C, a protein that binds to DNA, or D, an enzyme that synthesizes RNA? So what is a promoter? And I purposely chose this figure so that you didn't get the clue of what the promoter was. Okay, so the promoter is actually part of the DNA. Okay, so the DNA, so it's a specific sequence of DNA nucleotides. So it's that Tata box. And that Tata box is actually on the DNA. So it's A. And then finally, the final review question, portions of eukaryotic mRNA sequence that are removed during RNA processing are A, exons, B, caps, C, poly-A tails, or D, introns. And again, I chose the same figure because it doesn't give you the clues. So which of these four are removed during RNA processing? So remember, it's the intervening introns that get removed. The exons get expressed. The caps are there for translation. Poly-A tails prevent the thing from getting degraded. So the introns are the intervening, so they get spliced out. D. Okay, so we're done with the transcription part. So by now you should be able to explain the central dogma, as well as explain the main steps of transcription and describe how eukaryotic mRNA is processed. So in 8.4, we're going to get into now how that mRNA strand, it moves into the cytoplasm where translation takes place.